Instrumentation of FTIR. The main components of FTIR spectroscopy include infrared light source. A typical infrared light source can be a globar, a silicon carbide rod, which emits continuous radiation in the mid-infrared region, 4,000-400 centimeters. Beam splitter. A beam splitter divides the infrared light into two beams, one directed toward the sample and the other toward the reference. Interferometer. The interferometer, typically a Michelson interferometer, uses a moving mirror to modulate the light and create an interference pattern. This pattern is recorded and later converted into a frequency spectrum via Fourier transform. Sample holder. The sample is placed in the infrared beam, typically in the form of a thin film or in a gas cell for gases. Detector. Detectors like a DTGS, deuterated trig lysine sulfate, detector or MCT, mercury cadmium telluride, detect the intensity of the transmitted infrared radiation. Computer and software. The recorded interferogram is converted into a spectrum by applying a mathematical technique called Fourier transform, which translates the data from time domain to frequency domain. FTIR spectrum interpretation. The FTIR spectrum is typically presented as a graph of absorbance versus wave number, CM, where the wave number is inversely proportional to the wavelength. The spectrum shows a series of peaks corresponding to the vibrational frequencies of various functional groups in the sample. Some key points for interpreting FTIR spectra include fingerprints. The region of the FTIR spectrum below 1500 cm is called the fingerprint region and is highly specific to the compound being studied. This region can be used to identify compounds by comparing the spectrum to known references. Functional group region. The region above 1500 cm is typically used to identify specific functional groups in the sample, such as hydroxyl O carbonyl C equals O and amine NH groups. Applications of FTIR in environmental science: air pollution monitoring. FTIR is widely used to detect and quantify air pollutants like carbon dioxide CO, carbon monoxide CO, methane CH, nitrous oxide NO, sulfur dioxide SO, and volatile organic compounds VOX. It is used in both laboratory and field settings to monitor air quality and trace gases. Water quality monitoring. FTIR can be employed to analyze water samples for organic contaminants, including pesticides, herbicides, and other industrial pollutants. It is also used to identify and quantify organic compounds and pollutants in wastewater. Soil and sediment analysis. FTIR can provide insights into the composition of soil and sediment samples, especially for organic contaminants, humic substances, and pollutants. It helps to identify functional groups and molecular structures in organic matter present in these matrices. Identification of pollutants in environmental materials. FTIR is used for the identification and characterization of pollutants in environmental materials like sediments, plastics, and industrial waste. The technique is particularly valuable in assessing environmental contamination from industrial, agricultural, and urban sources. Biodegradation studies. FTIR can be used to study the degradation of organic pollutants in the environment, particularly in biodegradation studies where microorganisms break down contaminants. FTIR provides insights into the chemical changes occurring during the breakdown process. Analysis of greenhouse gases. FTIR is also used for the detection of greenhouse gases, such as CO, methane, and nitrous oxide, which are key contributors to global warming and climate change. Monitoring these gases helps to understand the effects of human activities on the atmosphere. Advantages of FTIR non-destructive analysis. FTIR is a non-destructive technique, meaning it does not alter or damage the sample, making it suitable for environmental monitoring where sample preservation is important. Wide range of applications. FTIR can analyze a wide range of materials, from gases and liquids to solids and powders, making it a versatile tool for environmental analysis. Speed and efficiency. FTIR provides rapid data acquisition and analysis. The ability to perform high-throughput analyses makes it suitable for large-scale environmental monitoring projects. High sensitivity. FTIR is capable of detecting low concentrations of pollutants, even in complex environmental matrices, providing high sensitivity for environmental monitoring. Limitations of FTIR water interference. Water has strong absorption bands in the FTIR spectrum, especially in the range of 3,000-3,600 centimeters, which can interfere with the analysis of aqueous samples. Sample preparation. In some cases, samples need to be prepared in a specific form, such as a thin film or KBR pellet, to be compatible with FTIR analysis. Quantitative analysis. While FTIR is excellent for qualitative analysis, quantitative analysis may be challenging in complex samples due to interference from other compounds or matrix effects. Previous year questions with answers. Which of the following techniques is used for the analysis of air pollutants like CO, CH, and NO? A. NMR. B. FTIR, C. XRD. D. Mass spectrometry. Answer. B. FTIR, UGC NET 2020. In FTIR spectroscopy, which region is typically used for the identification of functional groups in a sample? A. Fingerprint region. B. Above 1500 cm. C. Below 1500 cm. D. Below 1000 cm. Answer. B. Above 1500 cm. UGC NET 2021. What is the main advantage of FTIR spectroscopy in environmental analysis? A. High sensitivity. B. Non-destructive analysis. C. High resolution. D. Low cost. Answer. B. Non-destructive analysis. UGC NET 2019. Which of the following is a limitation of FTIR spectroscopy? A. Inability to detect organic compounds. B. Interference from water in the sample. C. Lack of sensitivity to volatile compounds. D. Only qualitative analysis is possible. Answer. B. Interference from water in the sample. UGC NET 2021. Which of the following pollutants can be detected using FTIR? A. Volatile organic compounds. VOX. B. Heavy metals. C. Radioactive substances. D. None of the above. Answer. A. Volatile organic compounds. VOX. UGC NET 2020. Conclusion. Fourier transform infrared spectroscopy (FTIR) is an essential tool in environmental science, widely used for identifying and quantifying pollutants in various environmental matrices. Understanding the principles, instrumentation, and applications of FTIR is crucial for UGC NET JRF environmental science candidates preparing for the exam. FTIR provides a non-destructive, rapid, and highly sensitive method for environmental monitoring, making it indispensable for both research and practical environmental analysis. You said, GCMS, ChatGPT said. GCMS, gas chromatography mass spectrometry, UGC NET JRF environmental science lecture. Gas chromatography mass spectrometry, GCMS, is a powerful and widely used analytical technique that combines the separation capabilities of gas chromatography with the detection power of mass spectrometry. GCMS is a crucial tool in environmental science, especially for the analysis of volatile and semi-volatile organic compounds in various environmental samples such as air, water, soil, and biological samples. This technique is highly effective in detecting pollutants, identifying organic contaminants, and studying complex environmental processes. Principle of GCMS. GCMS combines two techniques. Gas chromatography, GC. Gas chromatography is a technique used for separating volatile compounds in a sample. The sample is vaporized and injected into a chromatographic column, which is typically filled with a stationary phase, a liquid or solid material. As the sample moves through the column, different components are separated based on their interaction with the stationary phase and their volatility. The separated components exit the column and are directed into the mass spectrometer for further analysis. Mass spectrometry, MS. Mass spectrometry is used to detect and identify the separated components by measuring their mass to charge ratio, MZ. In this step, the ions are generated by ionizing the sample components, and their masses are measured. The resulting mass spectrum provides a fingerprint that can be used to identify the individual compounds in the sample. Each compound produces a unique fragmentation pattern in the mass spectrometer, which can be compared with reference databases to identify the chemical structure. GCMS instrumentation. The GCMS system consists of two main parts. Gas chromatograph GC. Injector. The sample is injected into the GC through the injector port. The sample is vaporized and carried by an inert carrier gas, e.g., helium or nitrogen, into the chromatographic column. Column. The column is the heart of the GC system, where separation of components occurs. It is typically coated with a stationary phase, liquid or solid, that interacts differently with each component based on its chemical properties. Detector. After separation, the components exit the column and are detected by the detector. The most common detector used in GC is the flame ionization detector, FID, or thermal conductivity detector, TCD, but for GCMS, the sample is directed into the mass spectrometer for further analysis. Mass spectrometer, MS. Ionization source. The separated sample components are introduced into the mass spectrometer, where they are ionized usually by electron impact, chemical ionization, or other ionization methods. Mass analyzer. The ionized molecules are separated based on their mass to charge ratio, MZ, using various types of mass analyzers, such as quadrupole, ion trap, or time of flight, TOF. Detector. The ions are detected and counted, producing a mass spectrum, which is recorded and analyzed by the system software. Steps in GCMS analysis. Sample preparation. The sample is prepared by extracting the analytes from the environmental matrix, e.g., soil, water, air, biological samples. The sample may need to be purified or concentrated before injection into the GC. Injection into GC. A small portion of the sample is injected into the gas chromatograph, where it is vaporized by the injector and carried by the carrier gas, e.g., helium, into the chromatographic column. Separation. As the sample moves through the column, the various components are separated based on their physical and chemical properties, such as boiling point, polarity, and interaction with the stationary phase. Detection by MS. After separation, the individual components are introduced into the mass spectrometer, where they are ionized. The mass spectrometer detects the ions based on their mass to charge ratio, MZ, and a mass spectrum is generated for each component. Data analysis. The mass spectra are analyzed by the software to identify the compounds present in the sample. The results are compared to known spectra in databases for identification. Applications of GCMS in environmental science. Environmental pollution monitoring. GCMS is widely used for the detection and quantification of volatile organic compounds, VOCs, and semi-volatile organic compounds, SVOCs, in environmental samples such as air, water, and soil. This is crucial for monitoring pollution levels and identifying contaminants from industrial, agricultural, and urban sources. Pesticide residue analysis. GCMS is extensively used to detect pesticide residues in food, water, soil, and environmental samples. The technique can identify and quantify multiple pesticides in a single analysis, making it highly effective for environmental safety and regulatory compliance. Analysis of water quality. GCMS helps to detect organic pollutants such as polycyclic aromatic hydrocarbons, PAHs, pesticides, herbicides, and industrial chemicals in water samples. This is crucial for ensuring safe drinking water and monitoring the contamination of aquatic ecosystems. Biomonitoring and toxicology. GCMS is used to study the bioaccumulation of contaminants in biological tissues, such as fish, plants, and humans. It is a valuable tool in environmental toxicology for tracking the effects of pollutants on living organisms. Indoor air quality studies. GCMS is used to monitor the presence of volatile organic compounds, VOCs, in indoor air, which can originate from building materials, cleaning products, and other sources. VOCs can contribute to poor indoor air quality and pose health risks. Forensic analysis. In environmental forensics, GCMS is used to trace the origin of pollutants and identify the specific chemicals responsible for contamination. This helps in identifying the sources of pollution and establishing liability. Greenhouse gas monitoring. GCMS is used to measure greenhouse gases such as methane, CH, nitrous oxide, NO, and carbon dioxide, CO, in the atmosphere. Used to measure greenhouse gases such as methane, CH, nitrous oxide, NO, and carbon dioxide, CO, in the atmosphere. This data is important for studying climate change and assessing the impact of human activities on global warming. Advantages of GCMS: high sensitivity and precision. GCMS is highly sensitive, capable of detecting trace levels of contaminants in complex environmental samples. It provides precise identification of compounds with minimal interference. Comprehensive compound identification. GCMS can analyze a wide range of organic compounds, from volatile gases to semi-volatile and non-volatile organic compounds, making it versatile for environmental analysis. Quantitative and qualitative analysis. GCMS provides both qualitative data identifying the compounds and quantitative data measuring the concentration of compounds, making it suitable for comprehensive environmental monitoring. Wide range of applications. The versatility of GCMS makes it applicable in various fields, from air and water quality monitoring to forensic analysis and environmental toxicology. Limitations of GCMS: sample complexity. GCMS may face difficulties in analyzing very complex matrices, such as highly contaminated or viscous samples, which may require extensive sample preparation. Limitations in analyzing non-volatile compounds. GCMS is primarily suited for the analysis of volatile and semi-volatile compounds. Non-volatile compounds may need to be derivatized before they can be analyzed by GCMS. High cost and maintenance. GCMS instruments are expensive and require regular maintenance, including calibration and the replacement of components such as a column and ion source. Previous year questions with answers. Which of the following techniques is commonly used for the detection of volatile organic compounds, VOCs, in environmental samples? A. GCMS. B. FTIR. C. HPLC. D. NMR. Answer: A. GCMS. UGCNET 2020. Which of the following is the main advantage of GCMS over other analytical techniques? A. Simplicity of instrumentation. B. Ability to identify and quantify both volatile and semi-volatile compounds. C. Low sensitivity. D. Limited application to organic compounds. Answer: B. Ability to identify and quantify both volatile and semi-volatile compounds. UGCNET 2019. In GCMS, which detector is used for the identification of compounds based on their mass to charge ratio, MZ? A. Flame ionization detector, FID. B. Mass spectrometer, MSC. Thermal conductivity detector, TCDT. Ultraviolet detector. Answer: B. Mass spectrometer, MS. UGCNET 2021. What is the primary use of GCMS in environmental science? A. To analyze the chemical composition of metals in water. B. To detect and quantify organic pollutants in air, water, and soil. C. To measure pH levels in soil. D. To assess the microbial content in wastewater. Answer: B. To detect and quantify organic pollutants in air, water, and soil. UGCNET 2020. Which of the following pollutants can be analyzed using GCMS? A. Heavy metals. B. Volatile organic compounds, VOCs. C. Radioactive isotopes. D. All of the above. Answer: B. Volatile organic compounds, VOCs. UGCNET 2019. Conclusion. GCMS is an indispensable analytical tool in environmental science for the detection, identification, and quantification of volatile and semi-volatile organic compounds in environmental matrices. Understanding the principles, instrumentation, and applications of GCMS is crucial for UGC NET JRF environmental science aspirants. This technique's sensitivity, precision, and ability to handle complex samples make it a go-to method for environmental monitoring and pollution analysis. You said, SEM. ChatGPT said. SEM, Scanning Electron Microscopy, UGC NET JRF Environmental Science Lecture. Scanning electron microscopy, SEM, is a powerful technique used to obtain detailed images of the surface structure of solid specimens. SEM is one of the most important tools in environmental science for examining the fine surface details of various environmental samples, including soil particles, pollutants, biological samples, and environmental materials like water and air filters. It provides high-resolution imaging and is used to study the morphology and composition of materials at the microscopic level. Principle of SEM. SEM works by scanning a focus beam of electrons across the surface of a sample. When the electron beam interacts with the surface, it produces secondary electrons, backscattered electrons, and X-rays. These signals are collected and converted into an image. The key principle behind SEM is the use of electrons, rather than light, to illuminate the sample. This allows SEM to achieve much higher resolution than traditional light microscopy. The main components of an SEM system include the electron gun, which produces the electron beam, the condenser lens, which focuses the beam, and detectors, which collect the emitted signals. The SEM is typically operated under a vacuum to prevent electron scattering by air molecules. SEM instrumentation electron source gun. The electron gun produces a focus beam of high-energy electrons that is directed onto the sample. Common sources of electrons include tungsten filaments or field emission guns. Condenser lens. The condenser lens. You said, SEM. ChatGPT said. SEM, Scanning Electron Microscopy, UGC NET JRF Environmental Science Lecture. Scanning electron microscopy, SEM, is a powerful technique used to obtain detailed images of the surface structure of solid specimens. SEM is one of the most important tools in environmental science for examining the fine surface details of various environmental samples, including soil particles, pollutants, biological samples, and environmental materials like water and air filters. It provides high-resolution imaging and is used to study the morphology and composition of materials at the microscopic level. Principle of SEM. SEM works by scanning a focus beam of electrons across the surface of a sample. When the electron beam interacts with the surface, it produces secondary electrons, backscattered electrons, and X-rays. These signals are collected and converted into an image. The key principle behind SEM is the use of electrons, rather than light, to illuminate the sample. This allows SEM to achieve much higher resolution than traditional light microscopy. The main components of an SEM system include the electron gun, which produces the electron beam, the condenser lens, which focuses the beam, and detectors, which collect the emitted signals. The SEM is typically operated under a vacuum to prevent electron scattering by air molecules. SEM instrumentation electron source gun. The electron gun produces a focus beam of high-energy electrons that is directed onto the sample. Common sources of electrons include tungsten filaments or field emission guns. Condenser lens. The condenser lens focuses the electron beam onto the sample. The degree of focus determines the resolution of the SEM. Scanning system. The scanning system moves the electron beam in a raster pattern back and forth across the sample surface. This ensures that the entire surface area is analyzed. Detector. Various detectors are used to collect the signals generated by the interaction of the electron beam with the sample. The most commonly used detectors include secondary electron detector, SED, detects low-energy electrons emitted from the surface, providing topographical information. Backscattered electron detector, BSED, detects high-energy electrons reflected from the sample, which provides information about the atomic number, contrast, and composition. Energy dispersive X-ray spectroscopy, EDS, detector, measures the X-rays emitted from the sample to identify and quantify elements present in the sample. Vacuum system. SEM operates in a vacuum to prevent the scattering of electrons by air molecules. The vacuum system maintains the required low-pressure environment. Sample stage. The sample is placed on a stage that can be tilted, rotated, and moved to allow imaging from different angles. The stage can also accommodate different sizes of samples. Working principle of SEM electron beam interaction. The electron beam is directed at the sample. The energy from the electrons interacts with the atoms of the sample, producing various signals, including secondary electrons used for imaging and backscattered electrons providing information about the sample's composition. Signal detection. The secondary and backscattered electrons are detected by detectors, and the signal is converted into an image. The intensity of the signal is related to the characteristics of the sample's surface, such as texture, roughness, and elemental composition. Image formation. The detected signals are displayed as an image on a computer screen. The image produced by SEM provides a detailed, magnified view of the surface structure of the sample. Energy dispersive X-ray analysis, EDS. In addition to imaging, SEM can be combined with EDS to analyze the elemental composition of the sample. When the electron beam strikes the sample, it causes atoms to emit X-rays. The energy of these X-rays is characteristic of the elements present in the sample, allowing for their identification. Applications of SEM in environmental science soil analysis. SEM is widely used in soil science to study the microstructure of soil particles. It provides detailed information about the shape, size, texture, and mineral composition of soil grains, which is crucial for understanding soil fertility, erosion, and contamination. Pollution studies. SEM can be used to examine the presence and distribution of pollutants in environmental samples. For example, SEM is used to study the accumulation of heavy metals and microplastics in sediments and biological organisms, providing insight into their environmental impact. Water quality monitoring. SEM is used to analyze water filtration systems, providing detailed images of the particles trapped in filters. It can also be used to study the microorganisms present in water, helping to assess water quality and pollution levels. Air pollution. SEM is used to analyze air pollution particles, including dust, soot, and aerosols. By examining their morphology and composition, SEM helps to understand the sources and behavior of air pollutants. Biological studies. In environmental microbiology, SEM is used to study microorganisms and their interactions with the environment. SEM allows researchers to observe the surface structures of bacteria, algae, and fungi in detail, which is important for understanding ecological processes like nutrient cycling and contamination. Environmental forensics. SEM is used in environmental forensics to investigate pollution incidents, identify contaminants in environmental samples, and trace the source of pollution. SEM is essential in forensic science to establish the origin of contaminants and aid in regulatory enforcement. Microplastic research. SEM is crucial in the study of microplastics, which have become a major environmental concern. SEM can provide high-resolution images of microplastic particles, helping to assess their distribution in marine and terrestrial ecosystems. Advantages of SEM high resolution. SEM can achieve resolutions up to 1 nanometer, allowing for the observation of fine surface details that cannot be seen with traditional optical microscopes. Surface imaging. SEM provides detailed information about the surface structure of materials, which is especially useful in environmental science for studying particles, pollutants, and biological samples. Elemental analysis. When combined with EDS, SEM provides elemental composition data, making it a valuable tool for environmental analysis and contamination studies. Non-destructive. SEM is a non-destructive technique, meaning that the sample is not altered or destroyed during analysis. This is important when studying rare or sensitive samples. Versatility. SEM can be used for a wide range of samples, from solids and powders to liquids with sample preparation and biological materials. Limitations of SEM sample preparation. Samples must be prepared by coating them with a thin layer of conductive material, e.g., gold or carbon, to prevent charging under the electron beam. Biological samples may require additional preparation, such as dehydration and fixation. Vacuum requirement. SEM requires a vacuum to operate, which can make it difficult to analyze samples that are sensitive to vacuum conditions, such as some biological specimens. Limited depth of field. SEM images have a limited depth of field, meaning that only a small portion of the sample can be in focus at any one time. This can be overcome by adjusting the sample or using advanced imaging techniques. Cost and complexity. SEM systems are expensive and require skilled operators to handle and maintain the equipment. Additionally, the analysis can be time-consuming and requires sophisticated software for data interpretation. Previous year questions with answers. Which of the following is a major advantage of scanning electron microscopy, SEM over light microscopy? A. Ability to observe live specimens. B. Higher magnification and resolution. C. No need for sample preparation. D. Ability to observe color in samples. Answer. B. Higher magnification and resolution. UGC NET 2021. In SEM, what type of detector is used to collect secondary electrons emitted from the surface of the sample? A. Backscattered electron detector. B. Secondary electron detector. C. X-ray detector. D. Ionization detector. Answer. B. Secondary electron detector. UGC NET 2020. Which environmental sample is most likely to be analyzed using SEM to study microplastics? A. Soil. B. Water. C. Air filters. D. All of the above. Answer. D. All of the above. UGC NET 2019. Which of the following techniques can be used in conjunction with SEM for elemental analysis of a sample? A. X-ray diffraction. XRD. B. Energy dispersive X-ray spectroscopy. EDS. C. High performance liquid chromatography. HPLC. D. Fluorescence microscopy. Answer. B. Energy dispersive X-ray spectroscopy. EDS. UGC NET 2020. In environmental studies, SEM is commonly used to analyze which of the following? A. Organic pollutants in soil. B. Particulate matter in air. C. Microorganisms in water. D. All of the above. Answer. D. All of the above. UGC NET 2019. Conclusion. Scanning electron microscopy (SEM) is a crucial technique in environmental science, offering high-resolution imaging and the ability to analyze the surface structure and composition of environmental samples. Its applications in pollution monitoring, soil analysis, microplastic research, and environmental forensics make it indispensable for researchers studying environmental contaminants. Understanding the working principle, instrumentation, and applications of SEM is essential for UGC NET JRF environmental science aspirants. You said, TEM. ChatGPT said. Transmission electron microscopy TEM UGC NET JRF environmental science lecture. Transmission electron microscopy TEM is another powerful imaging technique used to study the fine structure of materials at high magnifications. TEM offers an even higher resolution than scanning electron microscopy (SEM), allowing for the observation of internal structures at the atomic or molecular level. This technique is particularly valuable in environmental science for studying the microstructure of pollutants, biological samples, and environmental materials in detail. Principle of TEM. TEM works by passing a beam of electrons through a very thin sample. When the electron beam interacts with the sample, some electrons are transmitted through the sample, while others are scattered depending on the density and composition of the material. The transmitted electrons are then detected to form an image of the internal structure of the sample. The key principle behind TEM is the use of electrons instead of visible light, which allows for much higher resolution, enabling the study of materials at the nanometer or atomic scale. Unlike SEM, which detects secondary electrons emitted from the surface of the sample, TEM directly observes the electrons that pass through the sample, providing internal structural information. TEM instrumentation electron source gun. TEM uses an electron gun, usually a thermionic or field emission gun, to generate a beam of electrons. The electrons are accelerated using high voltage, usually in the range of 60 to 300 kV, and focused into a beam. Condenser lens. The condenser lens focuses the electron beam onto the sample. The condenser system can be adjusted to control the electron beam's intensity and size. Sample holder and thin sample sectioning. The sample must be very thin, typically less than 100 nanometers, so that electrons can pass through it. This thin sectioning is often done using ultramicrotomes or focused ion beams (FIB). The sample holder allows the specimen to be tilted and moved for different angles of analysis. Objective lens. The objective lens focuses the transmitted electrons onto the detector. This lens plays a crucial role in determining the resolution of the TEM. Imaging system. After passing through the sample, the transmitted electrons are focused onto a phosphor screen, charge-coupled device (CCD) camera, or film. This creates a high-resolution image of the internal structure of the sample. Detector. The image formed by the transmitted electrons is captured by the detector, which can either be a digital camera, a photographic plate, or an image recording system. Some advanced TEM systems also include energy dispersive X-ray spectroscopy (EDS) for elemental analysis. Working principle of TEM electron beam generation. The electron gun produces a high-energy electron beam, which is accelerated by high voltage. The beam passes through the condenser lens to focus the electrons onto the sample. Sample interaction. The electron beam interacts with the sample, passing through it or being absorbed by different areas based on the sample's density. The varying transmission of electrons creates contrast in the image. Image formation. The transmitted electrons are focused by the objective lens, forming an image on the detector. The contrast in the image is generated based on differences in sample thickness, density, and atomic number. Electron diffraction. In addition to providing images, TEM can also be used for electron diffraction. The diffraction patterns give information about the crystallographic structure of materials, helping to analyze the material's atomic arrangement. Energy dispersive X-ray spectroscopy (EDS). Many TEM instruments are equipped with EDS systems to analyze the elemental composition of the sample. When the electron beam strikes the sample, X-rays are emitted, and EDS detects the energy and intensity of these X-rays to determine the elemental content. Applications of TEM in environmental science study of pollution. TEM is used extensively in the study of pollution. It allows for the observation of fine particulate matter, such as soot, dust, and microplastics, at the molecular or atomic level. Researchers can analyze the size, shape, and composition of pollutants in air, water, and soil samples. Nanomaterials and nanoparticles. TEM is essential for the analysis of nanomaterials, which are often used in environmental remediation technologies. TEM can help identify the size, shape, and distribution of nanoparticles used in environmental cleanup efforts. Biological samples. TEM is commonly used to study the structure of biological samples, including microorganisms, viruses, and cellular structures. In environmental microbiology, TEM helps to understand the internal composition of bacteria, algae, and other microorganisms in aquatic environments. Soil and sediment studies. TEM is used to examine the fine structure of soil and sediment particles. By analyzing the internal structure of soil minerals, TEM provides insight into soil health, mineral composition, and the presence of pollutants such as heavy metals. Ecological studies. TEM is applied in ecological studies to investigate cellular and subcellular structures of plants, animals, and microorganisms. This technique provides high-resolution imaging of tissues and organelles, contributing to our understanding of environmental stress on biological organisms. Microplastics research. TEM is crucial for studying the structure and distribution of microplastics in environmental samples. It can reveal the size and morphology of microplastic particles, which is critical for assessing their environmental impact and understanding their movement through ecosystems. Wastewater treatment. TEM is used to investigate the effectiveness of filtration and purification systems. By observing the interaction of contaminants with treatment materials at a microscopic level, TEM helps to optimize wastewater treatment processes. Advantages of TEM high resolution. TEM provides much higher resolution compared to optical microscopy and SEM. It can achieve atomic level resolution, enabling the study of materials at the nanometer scale. Internal structural analysis. Unlike SEM, which provides surface information, TEM allows for the examination of the internal structure of the sample, making it ideal for studying cellular structures and nanoparticles. Elemental analysis. When coupled with EDS, TEM can also provide elemental composition analysis, enabling researchers to study the distribution of elements within the sample. Detailed imaging. TEM provides detailed images of biological samples, materials, and pollutants, helping to understand complex environmental processes at a molecular level. Versatility. TEM can be used for a wide range of samples, from biological specimens to environmental pollutants and nanomaterials. Limitations of TEM sample preparation. Sample preparation for TEM is time-consuming and requires the sample to be extremely thin, less than 100 nanometers. This can be a limitation for certain environmental samples that may be difficult to prepare in this way. Vacuum requirement. TEM operates under a vacuum, which can cause issues when analyzing delicate or volatile samples, such as water samples, which may need to be specially prepared. Complex operation. TEM is a complex instrument that requires skilled operators to prepare samples, operate the equipment, and interpret the data. It can be challenging for beginners to use effectively. Cost and maintenance. TEM machines are expensive, and their maintenance is highly specialized. This makes TEM a costly option for routine analysis, especially for smaller research institutions. Limited field of view. TEM typically provides a limited field of view compared to SEM. To observe large areas of a sample, multiple images must be captured and stitched together, which can be time-consuming. Previous year questions with answers. Which of the following is a major advantage of transmission electron microscopy, TEM over scanning electron microscopy, SEM? A. Higher resolution for surface imaging. B. Ability to observe the internal structure of samples. C. Easier sample preparation. D. Higher magnification of the sample surface. Answer. B. Ability to observe the internal structure of samples. UGC NET 2020. What is the primary disadvantage of transmission electron microscopy, TEM? A. Limited magnification. B. Inability to analyze internal structures. C. Complicated and time-consuming sample preparation. D. Requires no sample preparation. Answer. C. Complicated and time-consuming sample preparation. UGC NET 2021. Which of the following techniques can be coupled with TEM to identify the elemental composition of a sample? A. Energy dispersive X-ray spectroscopy. EDS. B. Fluorescence microscopy. C. X-ray diffraction. XRD. D. Gas chromatography. Answer. A. Energy dispersive X-ray spectroscopy. EDS. UGC NET 2019. In environmental studies, TEM is commonly used to analyze which of the following? A. The internal structure of microorganisms in water. B. The surface texture of soil particles. C. The morphology of air pollution particles. D. The distribution of contaminants in sediments. Answer. A. The internal structure of microorganisms in water. UGC NET 2021. Which of the following is a typical application of TEM in environmental science? A. Analyzing surface topography of pollutants. B. Studying the morphology and composition of microplastics. C. Measuring the electrical conductivity of materials. D. Identifying bacterial strains in soil samples. Answer. B. Studying the morphology and composition of microplastics. UGC NET 2020. Conclusion. Transmission electron microscopy, TEM, is an indispensable tool in environmental science for studying the fine internal structure of materials and pollutants. It offers exceptional resolution and the ability to analyze samples at the atomic scale, making it crucial for understanding environmental contaminants, biological systems, and nanomaterials. Mastery of TEM's principles, instrumentation, and applications is vital for UGC NET JRF environmental science aspirants aiming to excel in the exam. Environmental biology. Ecology is an interdisciplinary science. Ecology is the study of the interactions between organisms and their environment. Environmental biology. Ecology is an interdisciplinary science. Ecology is the study of the interactions between organisms and their environment, and it serves as a foundation for understanding how living organisms, including humans, interact with the planet. As an interdisciplinary science, ecology integrates principles from various fields such as biology, chemistry, physics, geography, and even social sciences. This broad-based approach allows ecologists to understand and solve complex environmental problems, considering the ecological, social, and economic aspects of human-environment interactions. It bridges the gap between theoretical and applied sciences, providing insights that are vital for addressing global challenges such as climate change, habitat loss, and resource depletion. Ecology is inherently interdisciplinary. Ecology is inherently interdisciplinary because ecosystems are complex and involve numerous variables that cannot be understood through the lens of a single discipline. For instance, understanding the impact of pollutants on a river ecosystem requires knowledge from chemistry to understand pollutants' chemical properties, biology to comprehend the effects on aquatic life, and physics to study water flow and dynamics. Ecology is inherently interdisciplinary because ecosystems are complex and involve numerous variables that cannot be understood through the lens of a single discipline. For instance, understanding the impact of pollutants on a river ecosystem requires knowledge from chemistry to understand pollutants' chemical properties, biology to comprehend the effects on aquatic life, and physics to study water flow and dynamics. Additionally, ecology intersects with geography in the study of ecosystems distribution across the globe, while it overlaps with social sciences in areas such as environmental policy, conservation, and sustainable development. This convergence of multiple fields makes ecology not just a branch of biology but a holistic science that can address environmental challenges on a global scale. Ecology also incorporates aspects of evolutionary biology to understand how species adapt to environmental changes and how these adaptations affect ecosystems. For example, the study of plant-pollinator interactions combines ecological principles with evolutionary biology, as it looks at how plants evolve traits to attract specific pollinators, which in turn influences the dynamics of the ecosystem. Similarly, the principles of genetics are integral in understanding population dynamics and genetic diversity within ecosystems, which are crucial for ecosystem stability and resilience. Furthermore, the study of human ecology examines how human activities such as urbanization, industrialization, and agriculture impact ecosystems. This is where the fields of sociology, economics, and environmental ethics come into play, as they help assess the social implications of environmental degradation and propose sustainable solutions. Understanding human behavior and its impact on natural resources is critical for creating policies and strategies that ensure the protection of biodiversity and the sustainable use of natural resources. Key areas of interdisciplinary ecology. Biogeochemistry. This area combines ecology, chemistry, and earth sciences to understand the cycling of nutrients and elements like carbon, nitrogen, and phosphorus in ecosystems. It helps explain how human activities affect nutrient cycles and contribute to issues like eutrophication or climate change. Global change ecology. It integrates ecology with atmospheric sciences and social sciences to study the effects of global climate change on ecosystems. This field assesses how shifts in temperature, precipitation, and other climatic factors influence biodiversity and ecosystem services, such as water purification, carbon sequestration, and pollination. Conservation biology. Combining principles from ecology, genetics, and environmental ethics, conservation biology aims to preserve biodiversity and ecosystems. It involves understanding the ecological and genetic factors that contribute to the decline of species and ecosystems and finding solutions to halt or reverse this loss. Environmental toxicology. This field brings together ecology, chemistry, and toxicology to study how pollutants affect living organisms and ecosystems. Ecologists study the concentration of pollutants in food webs and their long-term impacts on ecosystem health. Sociology and economics in ecology. Human ecology involves sociology and economics to understand how human communities use and manage natural resources. It helps design policies for sustainable resource management, urban planning, and pollution control. Key theoretical concepts in interdisciplinary ecology. Ecosystem services. The benefits that ecosystems provide to humans, including clean air and water, fertile soil, and pollination of crops. These services are studied through an interdisciplinary approach, as they involve biology, organisms providing services, chemistry, chemical processes, and economics value of these services. Biodiversity and ecosystem functioning. This area integrates evolutionary biology and ecology to understand how the diversity of species affects ecosystem processes like energy flow and nutrient cycling. It addresses questions such as how biodiversity contributes to ecosystem stability and resilience. Landscape ecology. A combination of ecology, geography, and environmental science. Landscape ecology studies spatial patterns in ecosystems. It looks at how habitat fragmentation, land use changes, and climate change affect the distribution of species and ecosystem services. Previous year questions with answers. Q1 2018. How does the concept of ecosystem services relate to environmental management? Answer. Ecosystem services are the direct and indirect benefits humans derive from ecosystems. Understanding these services, such as water purification, carbon sequestration, and pollination, is vital for effective environmental management. By quantifying these services, we can prioritize conservation efforts and develop sustainable management strategies. Q2 2019. Explain the importance of interdisciplinary approaches in studying global environmental change. Answer. Global environmental change, including climate change, deforestation, and biodiversity loss, involves complex interactions among biological, chemical, physical, and social factors. An interdisciplinary approach combines these aspects to develop comprehensive solutions. For example, climate change is not just a meteorological issue but also involves ecology, economics, and policymaking to mitigate its impacts. Q3 2020 discussed the role of human ecology in understanding environmental degradation. Answer. Human ecology studies the relationship between human populations and their environments. It examines how human activities, such as urbanization and industrialization, contribute to environmental degradation through resource overuse, pollution, and habitat destruction. This knowledge helps design sustainable development policies and mitigate negative human impacts on ecosystems. Q4 2021 describes the significance of biogeochemical cycles in ecosystem functioning. Answer. Biogeochemical cycles, such as the carbon, nitrogen, and water cycles, regulate the flow of essential nutrients and energy through ecosystems. They are vital for maintaining ecosystem balance and supporting life. Disruptions in these cycles, often caused by human activities, can lead to environmental issues like climate change, soil degradation, and eutrophication. Q5 2022 How does landscape ecology contribute to conservation strategies? Answer. Landscape ecology studies the spatial distribution of ecosystems and their connectivity. It helps in designing conservation strategies by identifying critical habitats, corridors for species movement, and areas vulnerable to fragmentation. Effective conservation requires understanding these patterns to maintain biodiversity and ecosystem services across large landscapes. Q6 2023 Explain the interdisciplinary nature of conservation biology and its importance in addressing biodiversity loss. Answer. Conservation biology integrates ecology, genetics, and social sciences to protect biodiversity. It studies the causes of biodiversity loss, including habitat destruction, overexploitation, and climate change. By combining these disciplines, conservation biologists can create strategies that address both ecological and socioeconomic factors contributing to species decline. These interdisciplinary approaches not only enhance our understanding of ecological processes but also guide the development of practical solutions to environmental issues. You said. Origin of life and speciation. ChatGPT said. Origin of life and speciation. The origin of life and the process of speciation are two fundamental concepts in evolutionary biology that are intertwined with the broader understanding of life on Earth. These topics explore the conditions that gave rise to life and the mechanisms by which new species emerge, contributing to the diversity of life we observe today. Origin of life. The origin of life is a subject of extensive research and debate, and while there is no single, definitive explanation, several hypotheses have been proposed to explain how life began on Earth. The most widely discussed hypothesis is the abiotic origin of life, which posits that life originated from simple organic compounds through chemical reactions that occurred in the primordial environment of early Earth. One key theory is the primordial soup hypothesis, proposed by Alexander Perrin and John Haldane in the 1920s. According to this theory, Earth's early atmosphere was rich in gases like methane, ammonia, and hydrogen, with little to no oxygen. Energy from lightning or ultraviolet radiation could have triggered chemical reactions among these simple molecules, leading to the formation of more complex organic compounds like amino acids, nucleotides, and lipids. These compounds, in turn, could have assembled into larger molecules, including proteins, nucleic acids like RNA, and lipid membranes, eventually forming protocells, which are considered the precursors to living cells. A key experiment that supported this idea was the Miller-Urey experiment, 1953, which simulated the early Earth environment in the lab. The experiment demonstrated that amino acids, the building blocks of proteins, could be synthesized from simple gases under conditions that mimic those of the early Earth. This provided evidence that organic molecules essential for life could form abiotically. Another important hypothesis is the RNA world hypothesis, which suggests that life may have originated from self-replicating RNA molecules. RNA has the ability to store genetic information and catalyze chemical reactions, which makes it a strong candidate for the first molecule of life. Origin of life and speciation. The origin of life and the process of speciation are two fundamental concepts in evolutionary biology that are intertwined with the broader understanding of life on Earth. These topics explore the conditions that gave rise to life and the mechanisms by which new species emerge, contributing to the diversity of life we observe today. Origin of life. The origin of life is a subject of extensive research and debate, and while there is no single, definitive explanation, several hypotheses have been proposed to explain how life began on Earth. The most widely discussed hypothesis is the abiotic origin of life, which posits that life originated from simple organic compounds through chemical reactions that occurred in the primordial environment of early Earth. One key theory is the primordial soup hypothesis, proposed by Alexander Perrin and John Haldane in the 1920s. According to this theory, Earth's early atmosphere was rich in gases like methane, ammonia, and hydrogen, with little to no oxygen. Energy from lightning or ultraviolet radiation could have triggered chemical reactions among these simple molecules, leading to the formation of more complex organic compounds like amino acids, nucleotides, and lipids. These compounds, in turn, could have assembled into larger molecules, including proteins, nucleic acids like RNA, and lipid membranes, eventually forming protocells, which are considered the precursors to living cells. A key experiment that supported this idea was the Miller-Urey experiment, 1953, which simulated the early Earth environment in the lab. The experiment demonstrated that amino acids, the building blocks of proteins, could be synthesized from simple gases under conditions that mimic those of the early Earth. This provided evidence that organic molecules essential for life could form abiotically. Another important hypothesis is the RNA world hypothesis, which suggests that life may have originated from self-replicating RNA molecules. RNA has the ability to store genetic information and catalyze chemical reactions, which makes it a strong candidate for the first molecule of life. According to this hypothesis, early life forms could have used RNA both for genetic material and for catalyzing the chemical reactions necessary for life processes. Over time, RNA molecules might have evolved into the more complex DNA-based life forms we see today. The hydrothermal vent hypothesis also offers a compelling explanation for the origin of life. This theory proposes that life could have originated in the warm, mineral-rich waters near underwater hydrothermal vents. These vents provide a stable environment and a rich supply of chemical energy, which could have driven the formation of complex organic molecules in the earliest forms of life. Although no conclusive evidence has yet been found to support any single hypothesis definitively, the study of the origin of life continues to be a dynamic field, combining insights from biology, chemistry, physics, and geology. Speciation. Speciation is the process by which new species arise from a common ancestral population. It is a central concept in evolutionary biology and explains how biodiversity evolves over time. Speciation occurs when populations of the same species become reproductively isolated from each other and diverge to the point where they can no longer interbreed, even if they come into contact again. There are two main types of speciation, allopatric and sympatric. Allopatric speciation. Allopatric speciation occurs when a population is geographically divided into two or more isolated subpopulations by physical barriers such as mountains, rivers, or glaciers. These isolated populations are exposed to different environmental pressures, which can lead to genetic divergence through mutations, natural selection, and genetic drift. Over time, these genetic differences accumulate, and the populations may evolve distinct traits that prevent interbreeding, even if the geographical barrier is removed. The classic example of allopatric speciation is the divergence of Darwin's finches on the Galapagos Islands. The finches on different islands were isolated from each other and evolved distinct beak shapes, sizes, and behaviors, adapted to the specific food sources available on their respective islands. This led to the formation of several new species of finches. Sympatric speciation. Sympatric speciation occurs when new species arise within the same geographical area, without any physical barrier separating the populations. This form of speciation is less common but can occur through mechanisms like polyploidy, sexual selection, and ecological niche differentiation. Polyploidy is a condition where an organism has more than two complete sets of chromosomes. In plants, polyploidy can result in reproductive isolation because the polyploid individuals cannot successfully interbreed with the original diploid population. Polyploidy is a common mechanism of speciation in plants, and many crop species, such as wheat and cotton, are polyploids. Sexual selection can also lead to sympatric speciation when individuals within a population begin to mate preferentially with others that have certain traits. Over time, this sexual preference can lead to reproductive isolation and the formation of distinct species. Ecological niche differentiation occurs when populations exploit different ecological resources or niches within the same environment. For example, if two populations of a species begin to specialize in different food sources or occupy different microhabitats, they may become reproductively isolated due to differences in behavior or timing of reproduction. Mechanisms of speciation. Several mechanisms drive the process of speciation. Genetic drift. Random changes in allele frequencies in small populations can lead to the accumulation of differences between populations, contributing to speciation. Natural selection. Environmental pressures such as climate, predators, or food availability can favor certain traits, leading to divergence in traits and reproductive isolation. Gene flow. The movement of genes between populations through migration can prevent speciation if gene flow is high. Reduced gene flow, however, can promote speciation. Key concepts in speciation. Reproductive isolation. This is a key feature of speciation. Populations must be reproductively isolated, unable to interbreed to form separate species. This can occur through pre-zygotic barriers such as different mating seasons or behavioral differences, or post-zygotic barriers such as hybrid infertility. Adaptive radiation. This is a form of rapid speciation where a single ancestral species rapidly diversifies into a wide variety of forms that adapt to different ecological niches. It is commonly observed when species colonize new environments with abundant resources and few competitors. Previous year questions with answers. Q1 2019. What is the significance of genetic drift in the process of speciation? Answer. Genetic drift refers to random changes in allele frequencies, which can be significant in small populations. Over time, these random changes can lead to genetic divergence between populations, contributing to speciation, especially in isolated populations where gene flow is limited. Q2 2018. Explain the role of polyploidy in plant speciation. Answer. Polyploidy occurs when an organism possesses multiple sets of chromosomes. In plants, polyploidy can lead to immediate reproductive isolation because polyploid individuals cannot mate with diploid individuals. This can result in the formation of new species, often seen in crop species. Q3 2020. Describe the process of allopatric speciation with an example. Answer. Allopatric speciation occurs when a population is geographically isolated, leading to reproductive isolation and genetic divergence over time. An example is the speciation of Darwin's finches on the Galapagos Islands, where populations on different islands evolved into distinct species due to isolation and different ecological pressures. Q4 2021. How do ecological niche differentiation and sexual selection contribute to sympatric speciation? Answer. Ecological niche differentiation occurs when populations exploit different resources or habitats, leading to reproductive isolation. Sexual selection can reinforce these differences by favoring mate choice based on specific traits, thus promoting speciation without geographical separation. Q5 2022. Discuss the significance of reproductive isolation in the formation of new species. Answer. Reproductive isolation is crucial in speciation as it prevents gene flow between populations. Without this isolation, populations can continue interbreeding and remain a single species. Mechanisms of reproductive isolation include behavioral differences, mating seasons, or physical barriers. Q6 2023. What are the key factors that drive the process of speciation? Answer. The key factors driving speciation include genetic drift, natural selection, reduced gene flow, and reproductive isolation. These factors can lead to genetic divergence between populations, eventually resulting in the formation of new species. The origin of life and speciation are central to understanding the diversity of life on Earth. While the origin of life remains an open question, the process of speciation provides a clear mechanism for how biodiversity evolves, driven by genetic, ecological, and behavioral factors. By studying these processes, we gain insights into the dynamics of evolution and the complexity of life's development on Earth. You said. Human ecology and settlement. ChatGPT said. Human ecology and settlement. Human ecology is the study of the relationship between humans and their environment, examining how human populations interact with their surroundings and how these interactions shape both the environment and human societies. Settlement, on the other hand, refers to the establishment of communities or habitation areas by human populations. These two fields are closely interlinked, as human settlements are influenced by ecological factors, and the way humans organize their settlements in turn affects the ecological system. Human ecology, an overview. Human ecology is a discipline emerged from the need to understand the ways in which human societies are shaped by their environments and how they, in turn, impact the natural world. The scope of human ecology covers various aspects, including population dynamics, resource utilization, environmental modification, and social structures. It integrates principles from biology, sociology, geography, anthropology, and environmental science to study the complex interactions between human beings and their environments. Human-environment interaction. Humans are not passive recipients of environmental conditions but actively shape and modify their surroundings. Historically, humans have altered landscapes through agriculture, urbanization, deforestation, and the construction of infrastructure. For example, the transition from hunter-gatherer societies to agricultural civilizations marked a significant environmental shift. Agricultural practices such as irrigation, land clearing, and monoculture farming have had profound impacts on soil fertility, water availability, and biodiversity. Adaptation and cultural practices. Human populations adapt to their environment through culture, technology, and social structures. These adaptations are often in response to local environmental conditions, such as climate, geography, and available resources. For instance, in arid regions, human settlements often rely on techniques like water conservation, rainwater harvesting, and the development of irrigation systems to survive and thrive. Similarly, in cold climates, humans have adapted by developing clothing, housing, and energy-efficient technologies. Ecological footprint. The ecological footprint is a key concept in human ecology that measures the environmental impact of human activities. It quantifies the amount of natural resources required to sustain human life, including energy, food, water, and waste disposal. Urbanization, industrialization, and population growth contribute to an increasing ecological footprint, leading to resource depletion, pollution, and climate change. Understanding this footprint is essential for sustainable development and managing human impact on the environment. Settlement, formation, and types. Human settlement refers to the establishment of communities or inhabited areas where people live, work, and interact. The location and structure of settlements are influenced by various ecological, social, and economic factors. Types of settlements. Rural settlements. These are sparsely populated areas, typically focused on agriculture, farming, and natural resource extraction. Rural settlements are often located in regions with fertile soil, access to water, and favorable climatic conditions for farming. Urban settlements. Urban settlements are densely populated areas where people live in cities or towns. They are centers of commerce, industry, culture, and education. Urbanization has been a global trend, particularly in the last century, due to industrialization, migration, and improved living standards. Suburban settlements. These settlements lie on the outskirts of cities. Suburban settlements. These settlements lie on the outskirts of cities and are often characterized by residential areas, low to medium population density, and proximity to urban centers. Suburban areas offer a balance between rural and urban living conditions. Ecological factors influence. Human ecology is a discipline emerged from the need to understand the ways in which human societies are shaped by their environments and how they, in turn, impact the natural world. The scope of human ecology covers various aspects, including population dynamics, resource utilization, environmental modification, and social structures. It integrates principles from biology, sociology, geography, anthropology, and environmental science to study the complex interactions between human beings and their environments. Human-environment interaction. Humans are not passive recipients of environmental conditions but actively shape and modify their surroundings. Historically, humans have altered landscapes through agriculture, urbanization, deforestation, and the construction of infrastructure. For example, the transition from hunter-gatherer societies to agricultural civilizations marked a significant environmental shift. Agricultural practices such as irrigation, land clearing, and monoculture farming have had profound impacts on soil fertility, water availability, and biodiversity. Adaptation and cultural practices. Human populations adapt to their environment through culture, technology, and social structures. These adaptations are often in response to local environmental conditions, such as climate, geography, and available resources. For instance, in arid regions, human settlements often rely on techniques like water conservation, rainwater harvesting, and the development of irrigation systems to survive and thrive. Similarly, in cold climates, humans have adapted by developing clothing, housing, and energy-efficient technologies. Ecological footprint. The ecological footprint is a key concept in human ecology that measures the environmental impact of human activities. It quantifies the amount of natural resources required to sustain human life, including energy, food, water, and waste disposal. Urbanization, industrialization, and population growth contribute to an increasing ecological footprint, leading to resource depletion, pollution, and climate change. Understanding this footprint is essential for sustainable development and managing human impact on the environment. Settlement, formation, and types. Human settlement refers to the establishment of communities or inhabited areas where people live, work, and interact. The location and structure of settlements are influenced by various ecological, social, and economic factors. Types of settlements. Rural settlements. These are sparsely populated areas, typically focused on agriculture, farming, and natural resource extraction. Rural settlements are often located in regions with fertile soil, access to water, and favorable climatic conditions for farming. Urban settlements. Urban settlements are densely populated areas where people live in cities or towns. They are centers of commerce, industry, culture, and education. Urbanization has been a global trend, particularly in the last century, due to industrialization, migration, and improved living standards. Suburban settlements. These settlements lie on the outskirts of cities and are often characterized by residential areas, low to medium population density, and proximity to urban centers. Suburban areas offer a balance between rural and urban living conditions. Ecological factors influencing settlement patterns. Water availability. Access to fresh water is one of the primary factors influencing the location of settlements. Historically, human settlements have developed along rivers, lakes, and coastlines where water is abundant. Climate, temperature, precipitation, and seasonal variations greatly influence settlement patterns. Areas with a temperate climate, moderate rainfall, and fertile soil are more likely to support permanent settlements. Topography. The physical characteristics of the land, such as mountains, plains, and valleys, affect where settlements are established. Settlements tend to develop in areas with favorable topography for agriculture, transportation, and defense. Soil fertility. Fertile soil supports agriculture, which is essential for sustaining human populations. Early civilizations, such as those in Mesopotamia, Egypt, and the Indus Valley, flourished in areas with rich, fertile soil for crop cultivation. Human settlement and environmental modification. The process of settling in a particular area often involves substantial modification of the natural environment. Early humans altered the landscape by clearing forests for agriculture, digging irrigation channels, and building shelters and roads. Over time, urbanization and industrialization have led to more profound environmental changes. Urban settlements require vast amounts of resources for infrastructure, energy, and housing. This can result in habitat loss, deforestation, air and water pollution, and changes in local microclimates. In recent decades, there has been a growing emphasis on sustainable settlement practices to mitigate environmental degradation. Concepts like green architecture, eco-friendly urban planning, and the use of renewable energy sources are becoming increasingly important in reducing the ecological impact of human settlements. Settlement evolution. The evolution of settlements has gone through several stages, from simple temporary shelters used by nomadic hunter-gatherers to complex urban societies with skyscrapers, roads, transportation networks, and utilities. In early human history, settlements were often small, isolated communities centered around agriculture or trade. Over time, settlements grew into cities, driven by population growth, trade, and the development of specialized occupations. The Industrial Revolution marked a significant turning point in settlement patterns, as mass production and mechanized agriculture led to the growth of cities and the decline of rural settlements. The modern era has seen rapid urbanization, with more than half of the world's population now living in cities. Urbanization and its ecological impact. Urbanization, the process of population movement from rural areas to urban centers, has led to significant ecological and environmental changes. While cities offer economic opportunities and improved standards of living, they also pose challenges to sustainability. Urban heat island effect. Urban areas tend to be warmer than the rural surroundings, a phenomenon known as the urban heat island effect. This occurs due to the increased absorption of heat by buildings, concrete, and asphalt, which traps heat and increases local temperatures. The heat island effect exacerbates energy consumption, particularly in the summer, as cities require more cooling. Pollution and waste management. The concentration of human activity in urban areas leads to higher levels of pollution, including air, water, and noise pollution. Industrial emissions, vehicle exhaust, and waste disposal contribute to the deterioration of urban air quality and water resources. Effective waste management systems, recycling, and sustainable waste disposal practices are essential to reduce the environmental impact of urbanization. Loss of biodiversity. As cities expand, natural habitats are cleared to make way for new buildings, roads, and infrastructure. This often leads to a reduction in biodiversity, as plants and animals lose their natural habitats. Urban sprawl can fragment ecosystems and create isolated pockets of nature, making it harder for species to survive and thrive. Sustainable urban planning. Sustainable urban planning focuses on creating cities that balance human needs with environmental preservation. This includes incorporating green spaces, efficient public transportation systems, renewable energy sources, and low-impact building materials. The goal is to reduce the ecological footprint of urban areas while enhancing the quality of life for residents. Previous year questions with answers. Q1 2018. What are the primary factors that influence human settlement patterns in relation to ecology? Answer. The primary factors influencing human settlement patterns include water availability, climate, topography, and soil fertility. Access to fresh water, moderate climate conditions, and fertile soil for agriculture are key ecological factors that shape the location and sustainability of settlements. Q2 2019. Discuss the environmental impacts of urbanization. Answer. Urbanization leads to several environmental impacts, including the urban heat island effect, increased pollution, air, water, noise, loss of biodiversity, and resource depletion. Urban areas require large amounts of energy, water, and land, which often result in habitat destruction and higher waste production. Q3 2020. How does the concept of ecological footprint relate to human settlements? Answer. The ecological footprint measures the environmental impact of human settlements, quantifying the resources required to sustain them, including food, water, energy, and waste disposal. Urban settlements, with their higher consumption of resources, tend to have a larger ecological footprint compared to rural areas. Q4 2021. Explain the role of sustainable urban planning in mitigating the environmental impact of cities. Answer. Sustainable urban planning involves designing cities that minimize environmental degradation by incorporating green spaces, renewable energy sources, efficient public transportation, and eco-friendly building materials. The goal is to reduce energy consumption, decrease pollution, and preserve biodiversity in urban environments. Q5 2022. What are the consequences of deforestation due to human settlement expansion? Answer. Deforestation caused by human settlement expansion leads to habitat loss, decreased biodiversity, altered water cycles, and increased carbon emissions. It contributes to climate change and reduces the availability of natural resources like timber and medicinal plants. Q6 2023. Discuss the impact of the urban heat island effect and how it can be mitigated. Answer. The urban heat island effect results in higher temperatures in cities compared to rural areas due to the absorption of heat by buildings and infrastructure. It can be mitigated by increasing green spaces, using reflective roofing materials, and incorporating urban vegetation to reduce heat absorption. Human ecology and settlement studies provide essential insights into the relationship between humans and their environment. By understanding these dynamics, we can better manage human populations, resources, and ecosystems to create sustainable communities that thrive both socially and ecologically. You said. Ecosystem structure and functions, structures, biotic and abiotic components. ChatGPT said. Ecosystem structure and functions, biotic and abiotic components. An ecosystem is a complex network of interactions between living organisms, biotic components, and their physical environment, abiotic components. It is a dynamic system where energy flows and matter cycles through various components, ensuring the survival and functioning of life forms. The structure and functions of an ecosystem are fundamental in understanding ecological processes and maintaining the balance of nature. Ecosystem structure. Ecosystem structure refers to the organization and arrangement of different components within an ecosystem, which include both biotic and abiotic components. These components interact with each other, creating the conditions necessary for life to thrive. Biotic components. Biotic components of an ecosystem are all living organisms that interact with each other in the environment. These components can be classified based on their role in the ecosystem. The primary categories of biotic components are producers, consumers, and decomposers. Producers. Autotrophs. Producers are organisms that can produce their own food using sunlight, in the case of plants, or inorganic chemicals, in the case of some bacteria. They form the foundation of the food chain and are responsible for converting solar energy into chemical energy, which is then passed on to other organisms in the ecosystem. Examples: plants, algae, and some bacteria, e.g., cyanobacteria. Consumers. Heterotrophs. Consumers are organisms that rely on other organisms for their energy needs. These can be classified into three main categories. Primary consumers, herbivores, animals that feed on producers. They are the first consumers in the food chain. Secondary consumers, carnivores, animals that feed on primary consumers. Tertiary consumers, apex predators, animals that are at the top of the food chain and have no natural predators. Examples: deer, primary consumer; lions, secondary consumer; and eagles, tertiary consumer. Decomposers. Saprotrophs. Decomposers break down dead organic matter, returning vital nutrients to the soil, water, and air. These organisms are essential for nutrient recycling in ecosystems. Examples: fungi, bacteria, earthworms. Abiotic components. Abiotic components are the non-living, physical, and chemical elements that influence the living organisms in the ecosystem. These components are crucial for the survival and functioning of the ecosystem, as they provide the necessary conditions for life processes. Light. Sunlight is the primary source of energy for most ecosystems. It drives photosynthesis, the process by which plants and algae convert solar energy into chemical energy. The amount and intensity of light influence the distribution and growth of organisms. Temperature. Temperature plays a vital role in determining the metabolic rates of organisms. Each species has an optimal temperature range for growth and reproduction. Extreme temperatures can limit the distribution of organisms. Water. Water is essential for all living organisms. It serves as a medium for chemical reactions, regulates temperature, and is necessary for the transport of nutrients and waste. The availability of water affects the distribution and abundance of organisms. Soil composition, texture, and nutrient content influence plant growth. The type of soil determines the kinds of plants that can grow in an area, which in turn affects the entire food web. Air, the composition of gases in the atmosphere, particularly oxygen and carbon dioxide, is crucial for respiration and photosynthesis. The concentration of gases can influence the functioning of the ecosystem. Minerals and nutrients. Soil and water contain essential minerals and nutrients like nitrogen, phosphorus, potassium, and calcium. These nutrients are crucial for plant growth and are transferred through the food chain. Ecosystem functions. Ecosystem functions refer to the various processes that take place within an ecosystem. These functions are vital for maintaining ecosystem stability, biodiversity, and sustainability. They include the flow of energy, cycling of nutrients, and various interactions that sustain life. Energy flow. Energy flows through an ecosystem in one direction, from producers to consumers to decomposers. This flow begins with the capture of solar energy by producers, e.g., plants through photosynthesis. This energy is then transferred to consumers when they feed on producers and subsequently to higher trophic levels. Energy is lost as heat at each trophic level due to metabolic processes. The total energy available decreases as it moves up the food chain, and hence ecosystems have a pyramid structure where producers have the most energy and apex predators have the least. Nutrient cycling. Nutrient cycling refers to the circulation of essential nutrients through the ecosystem. In ecosystems, nutrients like carbon, nitrogen, phosphorus, and sulfur are recycled through various biotic and abiotic components. Carbon cycle. Plants absorb carbon dioxide during photosynthesis and convert it into organic compounds. When organisms respire or decompose, carbon is released back into the atmosphere. Nitrogen cycle. Nitrogen in the atmosphere is fixed by bacteria into a usable form for plants. Herbivores consume these plants, and decomposers break down organic matter, releasing nitrogen back into the soil and atmosphere. Water cycle. Water evaporates from bodies of water, condenses in the atmosphere, and falls as precipitation, where it is absorbed by plants or returned to water bodies. Biological interactions. Ecosystems rely on a web of interactions between organisms, such as predation, competition, symbiosis, and mutualism. These interactions shape the structure and function of ecosystems. Predation. One organism predator feeds on another prey. Competition. Organisms compete for limited resources such as food, water, and shelter. Symbiosis. A close and long-term relationship between two species. It can be mutualistic, commensal, or parasitic. Mutualism. Both species benefit from the interaction, e.g., pollinators and plants. Commensalism. One species benefits, and the other is neither helped nor harmed, e.g., birds perching on trees. Parasitism. One organism benefits at the expense of the other, e.g., ticks on mammals. Regulation of population and community dynamics. Ecosystems maintain balance through various mechanisms that regulate the population of species. These mechanisms include predation, disease, competition, and resource availability. For example, predator-prey relationships help control the population size of species in an ecosystem. Waste recycling. Decomposers play a crucial role in recycling organic matter in ecosystems. They break down dead plants and animals, converting them into inorganic substances like carbon dioxide, water, and nutrients that are essential for plant growth. This recycling process ensures the continuous availability of nutrients for producers. Previous year questions with answers. Q1 2017. What are the biotic and abiotic components of an ecosystem, and how do they interact with each other? Answer. Biotic components include producers, consumers, and decomposers, which interact through feeding relationships and the transfer of energy. Abiotic components include sunlight, temperature, water, soil, and air, which provide the necessary conditions for life. These components interact through energy flow and nutrient cycling, ensuring the functioning and sustainability of the ecosystem. Q2 2018. Explain the process of energy flow in an ecosystem and its significance. Answer. Energy flows in an ecosystem from producers to consumers to decomposers. Producers capture solar energy through photosynthesis, which is passed on to consumers when they feed on plants or other animals. At each trophic level, energy is lost as heat due to metabolic processes. The energy flow is crucial for maintaining the balance and structure of the ecosystem. Q3 2019. Describe the nitrogen cycle and its importance in ecosystem functioning. Answer. The nitrogen cycle involves the transformation of nitrogen from the atmosphere into forms that can be used by plants and animals. Nitrogen fixation by bacteria converts atmospheric nitrogen into ammonia, which is then assimilated by plants. Herbivores consume plants, and decomposers break down organic matter, releasing nitrogen back into the soil and atmosphere. This cycle ensures the continuous availability of nitrogen, an essential nutrient for life. Q4 2020. What is the role of decomposers in an ecosystem? Answer. Decomposers break down dead organic matter, recycling nutrients back into the soil and water. This process ensures the continuous availability of essential nutrients for plants, which in turn supports the entire food chain. Without decomposers, ecosystems would accumulate dead organic matter, and nutrient cycling would halt, leading to the collapse of the ecosystem. Q5 2021. Explain how the ecological pyramid represents the flow of energy in an ecosystem. Answer. The ecological pyramid illustrates the distribution of energy among different trophic levels in an ecosystem. Producers form the base of the pyramid, with the highest amount of energy. As energy moves up the trophic levels to primary consumers, secondary consumers, and so on, it decreases due to energy loss at each level, primarily as heat through metabolic processes. The pyramid shape reflects this energy decrease. Q6 2022. Discuss the importance of the water cycle in maintaining ecosystem stability. Answer. The water cycle regulates the movement of water through the ecosystem, ensuring the availability of water for plants, animals, and microorganisms. Answer. The water cycle regulates the movement of water through the ecosystem, ensuring the availability of water for plants, animals, and microorganisms. It involves processes like evaporation, condensation, precipitation, and infiltration, which maintain moisture levels in ecosystems, regulate temperature, and support the growth of plants, thereby sustaining the entire ecosystem. By understanding the structure and functions of ecosystems, including the roles of biotic and abiotic components, we gain insights into how energy and matter flow through nature. This knowledge is essential for conserving biodiversity, managing natural resources, and promoting sustainable practices in the face of environmental challenges. You said. Functions, energy flow in ecosystems, energy flow models, food chains and food webs. ChatGPT said. Functions of ecosystems, energy flow models, food chains, and food webs. Energy flow within an ecosystem is a critical function that sustains life and ensures ecological balance. The flow of energy begins with the capture of sunlight by producers and continues through the various trophic levels via food chains and food webs. These processes not only facilitate growth, reproduction, and survival of organisms but also maintain the overall structure and function of ecosystems. Energy flow models help us understand how energy is transferred and how it influences ecosystem dynamics. Energy flow in ecosystems. Energy flow in an ecosystem refers to the movement of energy through the trophic levels, from the producers to primary, secondary, and tertiary consumers, and ultimately to decomposers. Energy enters ecosystems primarily through sunlight, which is captured by producers autotrophs in a process called photosynthesis. Sunlight is the primary source of energy. Solar energy is the foundation for most ecosystems. Producers such as plants, algae, and certain bacteria use sunlight to convert carbon dioxide and water into glucose and oxygen through photosynthesis. This glucose serves as a source of chemical energy for producers and other organisms in the ecosystem. Trophic levels. Producers autotrophs. These organisms produce their own food using sunlight or inorganic compounds. They occupy the first trophic level and form the base of the energy pyramid. Examples: plants, algae, phytoplankton. Consumers, heterotrophs. Consumers rely on other organisms for energy. These include herbivores, primary consumers, carnivores, secondary and tertiary consumers, and omnivores. Examples: deer, herbivore, lions, carnivore, and humans, omnivore. Decomposers, saprotrophs. Decomposers break down dead organic material, releasing energy back into the ecosystem and recycling nutrients. They play a crucial role in maintaining the flow of energy and nutrients. Examples: fungi, bacteria, and earthworms. Energy transfer. At each trophic level, energy is transferred to the next level through consumption. However, the transfer is inefficient, with about 90% of the energy being lost as heat due to metabolic processes. This energy loss at each level is why energy pyramids have a broad base of producers and a narrowing shape as it moves up to higher trophic levels. Energy efficiency. Typically, only about 10% of the energy at one trophic level is transferred to the next. This is known as the 10% rule of energy transfer. The rest is lost as heat due to metabolic processes such as movement, respiration, and digestion. Energy flow models. To understand how energy moves through an ecosystem, several models are used to represent energy flow and efficiency. These models simplify the complex interactions within ecosystems. The food chain model. A food chain represents a linear sequence of organisms in which energy is transferred from one organism to another. Each step in the food chain is called a trophic level, starting with producers and moving up through consumers. Example: Sunlight right pointing arrow grass producer right pointing arrow grasshopper primary consumer right pointing arrow frog secondary consumer right pointing arrow snake tertiary consumer. Energy flows in one direction from producers to apex consumers, with each trophic level representing a specific feeding relationship. The food web model. A food web is a more complex representation of the feeding relationships in an ecosystem. Unlike a food chain, which follows a single path, a food web consists of multiple interconnected food chains that reflect the diverse interactions between organisms. Food webs are more accurate models of energy flow because most organisms consume multiple types of food, not just one species. Example. In a grassland ecosystem, a rabbit might eat grass, but it may also be eaten by a fox. At the same time, insects like grasshoppers may eat the same grass, and birds might consume those insects. These interactions form a web of interconnected food chains. Food webs help to demonstrate the complexity and interdependence of species within an ecosystem. Food chains and food webs. Food chains. A food chain illustrates a linear path of energy transfer, shown to be human in an ecosystem. It starts with primary producers, plants, and moves up through various levels of consumers. Basic structure of a food chain. Producers, the base of the food chain, e.g., plants, algae. Primary consumers, herbivores that feed on producers, e.g., rabbits, insects. Secondary consumers, carnivores that feed on herbivores, e.g., frogs, snakes. Tertiary consumers, apex predators that feed on other carnivores, e.g., eagles, lions. Example. Grass right pointing arrow grasshopper right pointing arrow frog right pointing arrow snake right pointing arrow hawk. In each step of the food chain, energy is transferred, but a significant amount is lost as heat, meaning only a small portion is passed to the next level. Food webs. Food webs provide a more comprehensive and realistic model of energy flow. In an ecosystem, species rarely fit neatly into a single trophic level, and they often occupy different trophic levels at different stages of their life cycle. Food webs represent the multiple feeding relationships within a community. Example. In a forest ecosystem, trees producers provide food for herbivores like deer, insects, and birds. These herbivores may be eaten by carnivores like wolves, foxes, and birds of prey. The decomposers break down dead organisms and return nutrients to the soil, closing the cycle. Key characteristics of food webs. Complexity. Food webs contain numerous species and interconnected food chains. Energy transfer. Energy flows through the system in multiple directions, from producers to various levels of consumers. Resilience. Food webs are more resilient to changes in the ecosystem and simple food chains because of the redundancy of trophic interactions. If one species is removed, others may take its place in the web. Importance of energy flow in ecosystems. Energy flow in ecosystems is crucial for maintaining ecological balance and sustaining biodiversity. The efficiency of energy transfer impacts the abundance and distribution of species. Healthy ecosystems depend on the continuous flow of energy, and disruptions can lead to ecological imbalances, such as the collapse of species populations or shifts in community structure. Ecosystem productivity. The total amount of energy captured by producers in an ecosystem is called gross primary productivity (GPP). The energy available to consumers after producers use them for their own metabolism is called net primary productivity (NPP). NPP is a key indicator of an ecosystem's overall productivity and its capacity to support consumers. Trophic cascade. In a food web, changes at one trophic level can affect all other levels. For example, the removal of apex predators can lead to overpopulation of herbivores, which in turn depletes plant populations. This can alter the entire structure of the ecosystem, leading to a cascade effect. Previous year questions with answers. Q1 2016. Explain the 10% energy transfer rule in an ecosystem and its significance. Answer. The 10% energy transfer rule states that, on average, only about 10% of the energy at one trophic level is passed on to the next trophic level, while the rest is lost as heat. This rule highlights the inefficiency of energy transfer and explains why ecosystems have fewer apex predators compared to producers and primary consumers. Q2 2017. Differentiate between a food chain and a food web. Answer. A food chain represents a linear sequence of energy transfer, whereas a food web is a complex, interconnected system of multiple food chains. Food webs better reflect the complexity of real ecosystems, where organisms have multiple feeding relationships. Q3 2018. Discuss the role of decomposers in an ecosystem and how they contribute to energy flow. Answer. Decomposers break down dead organic material, releasing nutrients and energy back into the ecosystem. They recycle nutrients like carbon, nitrogen, and phosphorus, making them available for producers, which sustains the flow of energy through the food chain. Q4 2019. Explain how energy flow in an ecosystem is affected by human activities. Answer. Human activities like deforestation, pollution, and overfishing can disrupt the natural flow of energy in ecosystems. For instance, removing primary producers or apex predators can affect the entire food chain, leading to imbalances in species populations and nutrient cycling. Q5 2020. What is primary productivity, and how does it relate to ecosystem function? Answer. Primary productivity refers to the rate at which producers, e.g., plants, convert solar energy into chemical energy through photosynthesis. It is a critical measure of ecosystem function, as it determines the amount of energy available to consumers and thus the overall energy flow in the ecosystem. Q6 2021. What is the significance of the food web model in studying ecosystems? Answer. The food web model provides a more realistic representation of energy flow in ecosystems, illustrating the complex interactions between different species. It helps scientists understand ecosystem dynamics, the impact of species loss, and the resilience of ecosystems. By understanding energy flow, food chains, and food webs, we gain insights into the functional dynamics of ecosystems. These concepts are crucial for maintaining ecosystem health and biodiversity, making them essential for environmental science studies and conservation efforts. You said. Biogeochemical cycles. ChatGPT said. Biogeochemical cycles. An overview. Biogeochemical cycles refer to the movement of elements and compounds between living organisms and the non-living components of the earth, such as the atmosphere, soil, and water bodies. These cycles play a crucial role in maintaining ecological balance by recycling essential nutrients through various environmental processes. Without these cycles, essential elements like carbon, nitrogen, and phosphorus would not be replenished, and life on Earth could not be sustained. The major biogeochemical cycles that are essential to life include the carbon cycle, nitrogen cycle, phosphorus cycle, sulfur cycle, and water cycle. Each of these cycles describes the path through which elements move through the ecosystem, from abiotic components air, water, soil to biotic components organisms, and back again. 1. The carbon cycle. The carbon cycle describes the movement of carbon through the atmosphere, oceans, soil, and living organisms. Carbon is a vital element for all life forms because it forms the backbone of organic molecules such as carbohydrates, proteins, and lipids. Key processes. Photosynthesis. Plants, algae, and some bacteria absorb carbon dioxide (CO) from the atmosphere and convert it into glucose using sunlight. Respiration. Animals and plants release CO back into the atmosphere during cellular respiration. Decomposition. When organisms die, decomposers like fungi and bacteria break down their organic matter, releasing CO into the atmosphere. Fossilization. In some cases, organic matter is buried and forms fossil fuels, such as coal, oil, and natural gas. Combustion. The burning of fossil fuels releases large amounts of CO back into the atmosphere, contributing to global warming. Human impact. Human activities, such as deforestation and burning of fossil fuels, have significantly increased the concentration of CO in the atmosphere, leading to climate change and global warming. 2. The nitrogen cycle. The nitrogen cycle involves the transformation of nitrogen into various chemical forms that are utilized by living organisms. Nitrogen is a key component of amino acids and proteins, and it is required for the synthesis of DNA and RNA. Key processes. Nitrogen fixation. Nitrogen gas N in the atmosphere is converted into ammonia NH or related compounds by nitrogen-fixing bacteria, e.g., rhizobium found in the roots of leguminous plants or free living in the soil. Lightning also contributes to nitrogen fixation. Nitrification. Ammonia is converted into nitrites, NO, and then into nitrates, NO, by nitrifying bacteria in the soil. Nitrates are the most usable form of nitrogen for plants. Assimilation. Plants take up nitrates from the soil and incorporate them into proteins and other nitrogen-containing compounds. Animals obtain nitrogen by consuming plants or other animals. Ammonification. Decomposers break down organic matter in dead plants and animals, releasing ammonia back into the soil. Denitrification. Gentrifying bacteria convert nitrates back into nitrogen gas N, which is released into the atmosphere. Human impact. Human activities such as the use of synthetic fertilizers and burning of fossil fuels increase the availability of nitrogen compounds in ecosystems, leading to eutrophication, water pollution, and the creation of dead zones in aquatic environments. 3. The phosphorus cycle. The phosphorus cycle describes the movement of phosphorus through the environment. Phosphorus is essential for the formation of nucleic acids, DNA, RNA, and ATP, which are involved in energy transfer within cells. Key processes. Weathering of rocks. Phosphorus is primarily found in the form of phosphate minerals in rocks. Weathering breaks down these rocks, releasing phosphate ions co-cubed into the soil and water. Absorption by plants. Plants absorb phosphate from the soil and incorporate it into organic molecules. Animals obtain phosphorus by eating plants or other animals. Decomposition. When plants and animals die, decomposers break down their organic matter, returning phosphorus to the soil. Sedimentation. Phosphorus that is washed into water bodies can settle and become trapped in sediments, where it can remain for long periods. Human impact. The use of phosphate-rich fertilizers has increased phosphorus runoff into aquatic systems, contributing to nutrient pollution and eutrophication in water bodies. This leads to algal blooms and oxygen depletion, which can harm aquatic life. 4. The sulfur cycle. The sulfur cycle involves the movement of sulfur through the atmosphere, soil, water, and living organisms. Sulfur is an essential element in proteins and enzymes, and it is often found in the amino acids cysteine and methionine. Key processes. Atmospheric deposition. Sulfur compounds, such as sulfur dioxide, SO, and hydrogen sulfide HS, are released into the atmosphere through volcanic eruptions, fossil fuel combustion, and decomposition of organic matter. Sulfur oxidation. Sulfur dioxide can be oxidized to form sulfuric acid, HSO, contributing to acid rain. This acid can lower the pH of soils and water bodies. Absorption by plants. Plants absorb sulfur in the form of sulfate, squared from the soil. Decomposition. Decomposers break down sulfur-containing organic compounds, releasing sulfur into the soil and water. Human impact. Human activities such as burning coal and petroleum release sulfur dioxide into the atmosphere, leading to acid rain, which harms ecosystems, buildings, and human health. 5. The water cycle. The water cycle, also known as the hydrological cycle, describes the continuous movement of water on, above, and below the Earth's surface. Water is essential for all forms of life and plays a central role in the regulation of temperature and climate. Key processes. Evaporation. Water from oceans, rivers, lakes, and soil is converted into vapor by the heat of the sun. Transpiration. Water vapor is released by plants through stomata in their leaves. Condensation. Water vapor in the atmosphere cools and condenses into clouds. Precipitation. Water falls to the Earth's surface as rain, snow, or other forms of precipitation. Infiltration. Water is absorbed by the soil and replenishes groundwater supplies. Runoff. Water flows over the ground and returns to rivers, lakes, and oceans. Human impact. Human activities, such as deforestation, urbanization, and the extraction of groundwater, disrupt the natural water cycle. For instance, large-scale deforestation can reduce transpiration and alter rainfall patterns. Importance of biogeochemical cycles. Biogeochemical cycles are essential for the sustainability of ecosystems. They regulate the availability of nutrients, maintain ecological balance, and support the functioning of ecosystems. Human activities that disrupt these cycles, such as pollution and overexploitation of natural resources, can lead to environmental degradation, loss of biodiversity, and climate change. Previous year questions with answers. Q1 2015. Explain the process of nitrogen fixation and its importance in the nitrogen cycle. Answer. Nitrogen fixation is the process by which nitrogen gas N is converted into ammonia NH or nitrate snow by nitrogen-fixing bacteria. This process makes nitrogen available to plants, which is essential for protein synthesis and other biochemical processes. Q2 2016. Describe the role of decomposers in biogeochemical cycles. Answer. Decomposers break down dead organic matter, releasing nutrients like nitrogen, phosphorus, and sulfur back into the soil. These nutrients are then available for uptake by plants, ensuring the recycling of elements in the ecosystem. Q3 2017. How do human activities influence the phosphorus cycle? Answer. Human activities, particularly the use of phosphate fertilizers, increase the amount of phosphorus in water bodies. This excess phosphorus can lead to eutrophication, causing algal blooms and oxygen depletion, which harms aquatic ecosystems. Q4 2018. Explain the process of carbon sequestration and its significance in mitigating climate change. Answer. Carbon sequestration is the process of capturing and storing carbon dioxide (CO) from the atmosphere, often through the growth of plants or the storage of carbon in soils or geological formations. It helps mitigate climate change by reducing the concentration of greenhouse gases in the atmosphere. Q5 2019. Discuss the impact of sulfur deposition on ecosystems. Answer. Sulfur deposition, primarily from the burning of fossil fuels, can lead to acid rain, which lowers the pH of soils and water bodies. This can harm aquatic life, damage vegetation, and alter nutrient availability in ecosystems. By understanding biogeochemical cycles, we gain insight into how elements are recycled and how ecosystems function. These processes are critical for sustaining life and ensuring the resilience of ecosystems. Proper management of human activities that affect these cycles is essential for maintaining ecological balance. You said. Ecological succession. ChatGPT said. Ecological succession, an in-depth understanding. Ecological succession refers to the process through which ecosystems undergo a series of gradual changes over time, ultimately leading to a stable, mature community known as the climax community. This process is driven by the interactions between biotic, living organisms, and abiotic, non-living environmental factors components, as well as the disturbances that reset the ecological succession process. Ecological succession is a critical concept in understanding how ecosystems evolve, recover from disturbances, and maintain biodiversity. Succession can occur on different scales, such as the development of new ecosystems after a disturbance, secondary succession, or the formation of ecosystems from barren environments, primary succession. The two primary types of succession are primary and secondary succession. 1. Primary succession. Primary succession occurs in an area that has not previously supported life, such as newly formed volcanic islands, glacial retreats, or areas where soil is absent. The process starts from a bare, lifeless substrate, and it takes place in stages that involve the gradual accumulation of soil, vegetation, and animal life. This process is slow because it involves the creation of soil, which is essential for plant growth. Key steps in primary succession. Pioneer species. The first organisms to colonize a barren area are called pioneer species. These are typically hardy organisms such as lichens, mosses, and certain bacteria that can survive in harsh, nutrient-poor conditions. They help in breaking down rocks and creating small amounts of soil by weathering and the accumulation of organic matter. Soil formation. As pioneer species die and decompose, organic matter accumulates, and the soil begins to form. This makes the area more hospitable for more complex plant life. Invasion by new species. Once soil has accumulated, small herbaceous plants such as grasses and shrubs begin to take root. Over time, these plants alter the soil further by adding more organic matter and nutrients. Establishment of larger plants. As soil quality improves, larger plants such as trees can establish themselves, and a more complex plant community begins to form. Climax community. The succession process eventually leads to the establishment of a climax community, which is a stable, mature ecosystem. In many cases, this could be a forest or a grassland, depending on the climate and geographical conditions. Example. A classic example of primary succession is the colonization of a new volcanic island. Over time, the island is colonized by pioneer species, which help to build up the soil, allowing for more complex plant and animal life to establish. 2. Secondary succession. Secondary succession occurs in areas that have been previously inhabited but have experienced a disturbance such as a forest fire, logging, agriculture, or a flood. Unlike primary succession, the soil remains intact, and the area typically recovers more quickly. This is because the soil already contains the nutrients necessary to support plant growth, and some species may still be present in the environment, e.g., seeds, roots, or organisms. Key steps in secondary succession. Initial disturbance. A disturbance, fire, flood, or human activity leads to a reduction in the existing community, but the soil and some species remain. Colonization by weedy species. After the disturbance, weedy and fast-growing species, such as grasses and herbs, quickly colonize the area. These species are typically adapted to disturbed environments and can rapidly grow in the available space. Soil enrichment and invasion by new species. Over time, the presence of these early colonizers enriches the soil with organic matter and nutrients. This process facilitates the establishment of more diverse plant species, including shrubs and small trees. Mature vegetation and animal life. As the vegetation becomes more complex, animal life begins to recolonize the area. Larger trees and more complex plant species dominate the area, leading to the development of a mature community. Climax community. The final stage of secondary succession is the establishment of a climax community, which represents a stable, mature ecosystem. This could be a forest, grassland, or other community type, depending on environmental factors. Example. After a forest fire, the land may undergo secondary succession. Initially, grasses and small plants colonize the burned area, followed by shrubs and eventually large trees that restore the forest ecosystem. 3. Types of ecological succession. Autogenic succession. This type of succession occurs due to the internal factors of the ecosystem itself, such as plant growth and decomposition. Over time, the environment is altered by the organisms that live in it, making it more suitable for other species. Allogenic succession. Allogenic succession is driven by external environmental factors, such as climate change, weather patterns, or geological events. These changes influence the types of species that can survive and thrive in the area. 4. Factors affecting ecological succession. Climate, temperature, precipitation, and other climate factors play a significant role in determining the types of species that will colonize and thrive in an area. Soil composition. The availability of nutrients, pH, moisture, and texture of the soil influences the rate of succession and the types of plants that can grow. Biotic interactions, competition, predation, and mutualistic relationships among species can affect the progression of succession. Disturbances. Natural disturbances like fires, floods, and storms, as well as human-induced disturbances like logging and agriculture, can reset or influence the trajectory of succession. 5. Role of succession in ecosystem recovery. Ecological succession plays a critical role in ecosystem recovery after disturbances. Whether through primary or secondary succession, ecosystems gradually recover and restore biodiversity, soil fertility, and habitat structure. However, the process may take years to centuries, depending on the severity of the disturbance. Previous year questions with answers. Q1 2015. Define ecological succession and differentiate between primary and secondary succession. Answer. Ecological succession is a gradual process by which ecosystems develop and change over time. Primary succession occurs in areas without soil or organisms, while secondary succession occurs in areas where soil remains after a disturbance.